So we left last class talking about action potentials, how they're formed. The summation of graded potentials, they can either depolarize or hyperpolarize a nerve, and then when we reach threshold, voltage-gated channels open, and the sequence of events resulting in an action potential occur. And the action potential is the signal. That is what indicates there's something to sense, there's something to respond to, there's an action that needs to be taken. The action potential is what is transmitted through the body via the nerves to get from one point to another. And so we're confident in our ability to describe how an action potential is formed. We should be. What we want to do now is discuss how it moves, the propagation of the action potential. Because so far we've only discussed it in a bubble. We've been thinking one cell, the membrane potential is more negative inside than outside. We know that channels open and ions move, the cell depolarizes. But thinking in a bubble like that doesn't help us get from the trigger zone to the end of an axon. And it doesn't help us get from one axon or one neuron to another. So propagation of that signal is vitally important to get from a touch receptor in the skin to the cortex so that I know I'm touching the desk or holding a pen or my laser pointer. So we have to get from the trigger zone here all the way to the axon terminals in one neuron, and then we do that over and over again for every subsequent neuron in the loop or in the circuit. And we'll talk about circuits coming up. Now luckily, unlike graded potentials, we've talked about action potentials being all or nothing. They are on or they're off. They have the same strength regardless of where they are. If they fire, there is a certain magnitude or an amplitude to that action potential. And the same is true as you move from the trigger zone down the axon to the synaptic bulbs at the end. It's all or nothing. It's on or off. I also want to point out that this propagates in one direction only, from the trigger zone to the end, to the synaptic bulbs. It doesn't go backwards. And we'll illustrate that a little bit on the next slide. But I just want you to start thinking if one area has depolarized, it can't repolarize and then be active again for a brief period of time. We'll call that a refractory period. So we'll talk about that when we talk about the types of conduction that we have. Everything we've talked about so far is continuous. Everything we've talked about so far is continuous. If you take our one idea of a bubble and you link a few of those cells together, or different regions of an axon depolarize all at once, we're describing continuous conduction. And in continuous conduction, every part of the membrane must depolarize for the signal to be transmitted. So the trigger zone depolarizes, and that brings the immediate neighboring region to threshold, and that depolarizes. And then the neighboring region down here comes to threshold, and that depolarizes, and so on and so forth, all the way down the axon. The entire membrane depolarizes. And you see that shown here. The little green shaded areas are the leading edge of the action potential. That's where the sodium is rushing in. The voltage-gated channels are open. That's where the action potential is happening. And as you move from top to bottom, you see it progress further down the axon. So at the top, I have depolarization, and there's a couple little circles here. We'll talk about those in a second. But the immediate neighboring region depolarizes 4 milliseconds later, and the one after that, 5 milliseconds after that. Now, all those little circles are showing you are ionic current. Ionic current. Moving of sodium from the outside to the inside is an ionic current. Shows you the movement of ions. So think about that. If a voltage-gated channel opens and sodium rushes in, it doesn't just stay in some little pocket. It spreads out. The axon doesn't have a lot of sodium in it. So it rushes in and it immediately starts to move outwards. Sodium's positive. The charge right where it's entering is positive. And as it spreads that positive charge to the neighboring regions of the axon. So that's why the next region is close to threshold. When it reaches threshold, it depolarizes, and the chain reaction continues. This is a work-intensive kind of signal. It's slow. Every region of the membrane has to depolarize. We're activating sodium-potassium pumps the whole length of the axon. It's not very efficient. And that's why, largely in the white matter tracts, but in myelinated axons, conduction is so much faster because myelin insulates part of the axon so that it doesn't have to depolarize. This is called saltatory conduction. And the same progression is shown here from top to bottom. You have the same time frame for an action potential moving down a myelinated axon. But notice at every tier on the right-hand side, it's further down the axon. It's moved further. All right, that translates into a faster propagation speed. This is faster and a lot more efficient than continuous conduction. It's the same exact principle as continuous conduction. Myelin insulates part of the axon. There aren't any voltage-gated channels under the myelin. And so the depolarization skips between nodes. Remember the nodes of Ranvier? Little spaces between the marshmallows on an axon, the spaces between the myelin. Those can depolarize, absolutely. But the myelinated areas can't. And so we get depolarization in one node, followed by the next, and every node after. So it leapfrogs each of these nodes, resulting in a much faster conduction. And you see the same exact ion currents happening. Sodium still rushes in, it still spreads out, but it doesn't leave the membrane, it doesn't affect depolarization until much further down the axon. The current flows inside the cell when it reaches the node, triggering depolarization, allowing it to jump between nodes. Much faster conduction. Also more efficient because you only have sodium-potassium pumps working at these nodes. Fewer pumps break down less ATP, generally a more energy efficient form of communication. So those are the two main benefits of saltatory over continuous. Faster, more efficient. And that's the major defining factor for propagation speed or signal speed. But there are some other factors that affect speed as well. The diameter of the axon will affect how quickly the membrane depolarizes and how quickly that signal is sent. The larger the diameter, the larger the surface area, and the more opportunity you have to move ions. If we can move ions quickly, that means we can transmit the signal more quickly as well. So the larger diameter axons will result in a faster conduction speed. It seems a little counterintuitive. You might be thinking, well, there's more membrane to depolarize. That's not the case. More opportunity to move ions allows faster communication speed. What we just discussed is the major factor defining propagation speed, the amount of myelin. More myelin, more saltatory, faster conduction. And of course, temperature. part of the reason why icing an injury might help to minimize or numb the area or numb the sensation of pain. All chemical reactions. The speed of all chemical reactions is proportional to temperature. So ion movement, sodium, potassium pumping, all happen faster when warm, slower when cold. So signal transmission is faster when warm, slower when cold. Notice that nothing here implies there's a difference in signal strength. We talked about this already, the idea that action potentials can't increase in magnitude, they're fixed. And the only way that we can increase the intensity of an action potential is to fire it more quickly. So these factors don't change the strength of the action potential, they change how quickly the action potential fires or is transmitted. And this is why we can differentiate between light touch and firm touch. Firm touch, action potentials are being fired much more quickly in succession, more of a signal. Or when we talked about the negative feedback loop of blood pressure. Remember those baroreceptors way back when? They fire more quickly with high blood pressure, more of a signal. The size, the amplitude is not affected. So if we're comfortable with how an action potential is triggered and that it moves down the axon, what I want to focus on now is how it moves from one neuron to another. We've already introduced the idea of a synapse where neurons are connected. But they often, the majority of the time, they don't physically touch. There's a small space between them. So how is it that the signal can get from one neuron to another? How is the signal transmitted at a synapse? So synapse is simply the junction where two neurons connect. And normally the axon connects to dendrites. The incoming or presynaptic neuron connects to the dendrites of the outgoing or the postsynaptic neuron. And this is, we're articulating here a difference that we've already we've talked about. The postsynaptic potentials. When we talked about graded potentials, that indicates that it's in the downstream neuron that those occur. The incoming signal is in a presynaptic neuron, the outgoing signal in a postsynaptic neuron, and this is asterisked because it only works if there are two neurons that are connected. If one neuron is transmitting a signal to a muscle cell or a gland, it's not a postsynaptic neuron. It's a postsynaptic cell in that case. If you think about a basic relay with neurons in series, the presynaptic connects to the postsynaptic and the signal is transmitted all the way down. Now there are two ways that signals can be transmitted. First one is less common. When neurons physically touch or cells are physically connected, gap junctions, remember those from way back when? Will allow the potential to spread between cells. And really that simply means it allows sodium to spill from one cell to the next, raising membrane potential closer to threshold, allowing depolarization. So an electrical synapse is a direct connection of two cells where the signal is transmitted via gap junctions, sodium moving through gap junctions. This happens a lot in smooth and cardiac muscles, areas where it's really important to have no disruption in the signal or delay in the signal. You wouldn't really want part of your heart to not contract during a heartbeat, right? You want it all to be orchestrated and in unison. So there are a lot of these types of connections in cardiac muscle. So benefits here are it's pretty fast, and it allows uniform conductance. All the cells that are connected will essentially depolarize at once. And they do it really quickly because of the gap junctions. The more common form that we see in the peripheral nervous system between neurons and muscle tissue, between neurons and glands, is a chemical synapse, where there's actually a space. The neuron is not connected to the tissue, connected to the cell, and so something has to jump across that space from the presynaptic to the postsynaptic neuron to carry the action potential signal. We've seen these before. These are neurotransmitters that make that leap. And we'll discuss the chemical synapse in depth. I have another really nice series of animated slides that illustrate this process, courtesy of Matt. We'll take a look at those quickly or shortly. I just want to show you the types of synapses and how they might vary, because so far our thinking's very linear. Cell body, axon, to dendrites of the next nerve, to the cell body, to the axon, to dendrites of the next neuron, etc. And that's one of the most common types of synapses. The axodendritic synapse in the top right. Axodendritic. The axon connects to the dendrites. But, as we'll see, we'll look at some more complicated circuitry at the end of the lecture. And we'll gradually build on this simple idea of action potentials and propagation. But neurons can synapse in many different places on the postsynaptic neuron. We have an axosomatic connection where the presynaptic neuron connects directly to the body of the postsynaptic cell, or the axoaxonic connection, where the axon connects directly to the trigger zone. And everything in between can happen too. In fact, one neuron can touch or connect to many downstream postsynaptic neurons all at once. You can have thousands of presynaptic neurons attaching to this one cell body. Thousands, tens of thousands. The network gets ever more complex, which is good because it allows ever more complex action and higher order thinking and consciousness. This is really the basis for how you're able to sit there and decide to pay attention or not, or what to write down or not, or make a choice to go have a sandwich or pizza later. It's all just the complexity of these neural circuits. And some gut bacteria, but we'll talk about that later. Or we have already. So myriad of different kinds of synapses, the three main classes shown here. They can reinforce, they can interrupt the graded potentials, but I wanted to point this out for you. Let's focus just on the axodendritic, the classic example of a synapse. And I'll do this quickly in this other series of slides. You have this figure. We're zooming in here on one terminal end bulb. There are many of these that branch off of one axon. And these end bulbs will connect to the postsynaptic neuron either on the dendrites or the body or on the trigger zone. And each one of these end bulbs looks something like this. It looks pretty complicated. We're going to go through in depth what happens to illustrate how we can get across this space. And the space that I'm referring to in question is called the synaptic or synaptic cleft. The presynaptic neuron here with its end bulb, the postsynaptic neuron on the bottom, they don't actually touch. Yet the signal, the action potential, has to get across. It can't jump. So something has to transmit that signal. So let's move in order. And you can follow along with the numbers on this figure. We're going to go through number 1 through I guess 5, no, we'll go through 7. Yeah, we'll go number 1 through 7. We have an action potential arriving at the end bulb, as shown here. Unidirectional, sequential activation, depolarization reaches the end terminal bulb. When that happens, we've relied so far on voltage-gated sodium channels to relay the action potential. But when we reach the end, a different kind of voltage-gated channel comes into play. Voltage-gated calcium channels, not potassium. Voltage-gated calcium channels open up, and calcium rushes in. We will see as we go through the course that calcium is one of the early warning signals. It's the alarm bell to initiate some action in the cell. And I always default back to muscle. Calcium is the thing that initiates contraction. The reason I can move and contract a muscle is calcium told the muscle to do that. Here, calcium does something different, but it initiates a major action in the end bulb. Calcium binds to or turns on and prompts these vesicles to move towards the membrane. And the vesicle is just a pocket. It's almost like a mini cell within a cell. And these pockets are full of neurotransmitter. In this case, it's going to be acetylcholine, but there are a myriad of neurotransmitters. And so calcium tells these little pockets of neurotransmitter, move to the membrane and release that neurotransmitter. Those little pockets fuse with the membrane and they open up. They release that neurotransmitter into the synaptic cleft. They release it. It's outside of the cell. But importantly, it's right in the vicinity of the ligand-gated channels of the postsynaptic nerve. And so those little black dots have bound to these yellow squares, which are the ligand-gated channels on the postsynaptic neuron. And what does that cause? Those ligand-gated channels open. Acetylcholine-gated channels are often sodium channels, so sodium rushes in. And you can see where this is going. Sodium rushes in. Membrane potential depolarizes. If it reaches threshold, an action potential occurs and is transmitted down the plasma membrane, down the membrane of the axon. We'll assume this all happened in the trigger zone, and this is it's labeled as a graded potential. We'll assume that the graded potentials summarized to reach threshold and create an action potential, and that is moving down the axon. I jumped way ahead. You get the idea. If enough sodium rushes in, and there's enough of a depolarizing graded potential so that it reaches threshold, an action potential occurs. That is an example of an excitatory postsynaptic potential, an EPSP. Depolarized the membrane, brought the potential closer to threshold, and eventually makes an action potential. You've been following along here. You can see the same cascade, the same sequence of events that result in, eventually, a nerve impulse, which is another word for an action potential moving forward. Brief note while we're here and I'm thinking about it. I haven't yet done it, but these slides will be posted to the class Moodle. If you were looking for them yesterday, I don't think they're on the lab Moodle page yet, and they're not going to be, just so we're not confused about what material is meant for the midterm or the exams or who knows what. I'll put all of these on the class Moodle page, but they're not up there yet, so don't fret, they will be. Okay? So an excitatory postsynaptic potential, we know, to be one that causes the membrane to depolarize. the same sequence of events can occur but elicit an inhibitory postsynaptic potential. Maybe instead of acetylcholine, the presynaptic neuron released glycine. Or maybe instead of acetylcholine, the presynaptic neuron released something we'll see today, referred to as GABA. GABA is a major inhibitory neurotransmitter. Or maybe the ligand-gated channel allowed potassium out. All of those things would hyperpolarize and inhibit the postsynaptic neuron. Inhibitory postsynaptic potential. And this probably sounds repetitive, and it's supposed to be. We've seen these terms before because we've come full circle. When this action potential is transmitted across the synapse, it becomes a graded potential for the next neuron. And the whole cycle begins again. We can see an increase in the stimulus strength for the graded potential, or we can see the summation of the graded potential to cause the nervous impulse to be relayed. And here's the kicker. We've been thinking in very linear serial terms. One neuron connects to one more, and so on down the chain. But one neuron can connect to 1,000 neurons, or 1,000 neurons can converge on one neuron itself. And so there can be 1,000 IPSPs or EPSPs or a mix of the two that all influence the postsynaptic neuron. And this is an important next step to start wrapping your head around. Transmission of an action potential is not so simple. Processing, being aware, not a simple process. We need these complex circuits and complex responses to support that higher order thinking. Now, I alluded to in that example the idea that the postsynaptic signal can vary depending on the neurotransmitter, depending on the channel that's open. And sometimes even the same neurotransmitter can cause the opposite signal, but I don't want to get into that in too much depth to confuse you anymore. But the amount of channels, the types of neurotransmitters all dictate what the net effect is on that downstream neuron. Is it a net excitatory signal? Is it a net inhibitory signal? And so I just want to explore briefly some of the flavors of neurotransmitter and some of the flavors of channels on that postsynaptic membrane. There are two main types, ionotropic and metabotropic. Everything we've talked about so far is ionotropic. The receptor and the channel are one and the same. So if a ligand binds to a channel and then the channel opens, ionotropic. If they're not the same, it's what we call metabotropic, and I'll show you an example on this slide. This is very similar to another image that you've seen that we presented earlier in these slides. In fact, the very first receptor at the top is one we've seen before. This is a ligand-gated channel where acetylcholine binds to the channel, and once it's bound, the channel opens up. This is the ligand-gated channel we described in the animation. This is the one that is responsible for an excitatory graded potential in the postsynaptic neuron because sodium rushes in. Sodium here, the green dots, rushes in. complementary type of channel is shown in the second series of graphs. This is one that responds to gamma aminobutyric acid is the full name, but we short form it as GABA. And if acetylcholine is the neurotransmitter that turns things on, GABA is the neurotransmitter that turns things off. GABA is the main inhibitory neurotransmitter in our nervous system. Acetylcholine activates, GABA inhibits. GABA also works through an ionotropic channel. It binds, and you can see here chloride rushing in. Chloride's negative, more negative inside, means the membrane potential goes down, it hyperpolarizes, it's away from threshold, so it's inactivated or turned off or turned down. Inhibited. The last example is a metabotropic receptor where the transmitter doesn't bind to the channel. So you can see these two are physically separated. In this example, it also uses acetylcholine, so we're adding another layer of complexity, illustrating how these circuits can be more complex and give rise to different downstream stimuli. If the receptor binds acetylcholine, the channel doesn't open right away. It usually initiates what we call a signaling cascade. A signaling cascade. These are often connected to things that are called G proteins, and we don't need to go into depth about what those are. You'll learn about them in biochemistry and metabolism. But it turns the G protein on, and that turns something else on, and there's a series of events that results in the channel opening. Actually, probably one of the better examples is insulin. I don't know how familiar you are with the insulin signaling cascade, but insulin binds to a receptor in the cell and doesn't open channels for glucose right away. There are many complicated overlapping steps that result in channels opening up and glucose moving into the cell. That's probably one of the more common ones. But there are many steps. We're not going to go through that in detail. So what's the point of showing you all this complicated information? This really allows nuance in the nervous signal, in transmission of nervous signals. We can have robust, large, positive activation. We can delay the signal and have a positive signal happen in quick succession. Depending on the types of receptors, maybe the signal is tempered. Maybe we have to turn something off, like is the case with reciprocal innervation in the reflexes we discussed. There's a lot of nuance and color and context that we can add to these neural circuits, depending on the type of neurotransmitter, depending on the channel, depending on the number of incoming signals. Now what happens? All of this depends on there being no neurotransmitter there initially. If we want the signal to be sent between two neurons across a synaptic cleft, it assumes that there's none there initially. So after the signal's been sent, we need to remove it. If the neurotransmitter stays in that space, it'll just keep activating the receptor and ion channels. And so if we only want to move our arm a little bit, that's a bad thing. We don't want to always activate the biceps muscles and then throw our glass of water behind us. We need to remove that signal. And so we can remove the neurotransmitter by any one of three ways, or the combination of three ways. First way is the neurotransmitter naturally will diffuse. Like sodium, like any ion, it will move from high to low concentration. when it's released from the presynaptic neuron, it's in high concentration in the cleft. So it will move out of the cleft. It will move out of the cleft into the extracellular space, into the blood. Who cares? It's not in the cleft. It moves away. Number two, we can degrade it. There are specific proteins nearby, just like the receptors are proteins, that bind and degrade the neurotransmitter. And so our most popular neurotransmitter is acetylcholine. Acetylcholine esterase is an enzyme that breaks it down. And you'll almost always, this is a hint for biochem, you'll be able to tell when something is cleaved or cut or broken if it has A's at the end. Acetylcholine ester A's, something's chopped off. Sodium, potassium, ATPase, phosphates chopped off, right? So enzymes break down the neurotransmitter, or it's recycled. There are pumps on the presynaptic neuron that will take it back up because it doesn't want to run out. It wants to store that neurotransmitter again in vesicles that will allow it to send another nervous signal. If it doesn't, there's no neurotransmitter, it can't do that, right? The point of having a synapse is lost. So there are pumps that will move the neurotransmitter back into the cell, or the neuroglia might help get rid of it, the supporting cells in the nervous system. So if we do this successfully, we've delivered the signal, and we're ready to deliver a new signal. If we do this 1,000 times, the postsynaptic neuron needs to take in and integrate all of those signals. And this, like we talked about before, is the process of summation. So graded potentials are summated. If two excitatory potentials overlap, they have a greater effect than either in isolation. From this graph, or the figure that we saw already last class. So imagine stimulus 1, stimulus 2, stimulus 999, stimulus 1000, stimulus 5446, all of these upstream presynaptic nerves adding together or subtracting away, making some net graded potential in the postsynaptic nerve. Summation is the process of integrating. And you think about the cortex integrating information to come up with a plan, to execute an action. It's the summation of all the incoming information that we're talking about. So summation, the process by which graded potentials add together or subtract if they're inhibitory. Summation in one postsynaptic neuron can be of two forms. It's either positive or negative, but it can happen in two ways. The first way is spatial summation. Spatial summation. So we can get two excitatory graded potentials from different locations. So here I have two presynaptic neurons in blue, one postsynaptic neuron in red. And if these fire, if the signal is being transmitted from blue to red, I can either have neuron 1 activate and excite the downstream nerve on its own, by itself. It doesn't depolarize the membrane completely. It brings it towards threshold, but it never reaches threshold. And then it dies off. And the same with presynaptic neuron number 2. Each of these will excite, but not trigger the postsynaptic neuron. If they fire independently, they never trigger the postsynaptic neuron. But what if they both fired at the same time? This is a depiction of what we saw in the last slide. Neuron 1 and neuron 2, the presynaptic neurons both depolarize, both deliver action potential simultaneously. And so the EPSP is additive, and we reach threshold in the postsynaptic neuron. Spatial summation. Summation from 2 different locations. Well, we can also then have temporal summation. If we follow the train of thought, this is not due to signals from multiple locations. Now this is an increase in the frequency, or different signals at different times, put it that way. Signals from the same location, but at different times. So this is our typical linear neural circuit. 1 presynaptic neuron in blue, 1 postsynaptic neuron in red. Let's say the presynaptic neuron fires once. Well, you already missed it. It fired, membrane potential went up, but didn't reach threshold. Okay, well maybe it fires again. Same thing. Too slow. But what if it fires faster? What if it fires 5 times, 4 times? Every action potential builds. The graded potential builds in the postsynaptic neuron, and eventually we reach threshold. Temporal summation. And this is about as complex as the book wants to go with it, and I think this is a good place to not stop lecture, but to end our extrapolation of this idea. Thousands of neurons, tens of thousands of neurons wouldn't fit on the slide, but you get the idea. There might be multiple inputs that are excitatory, multiple inputs that are inhibitory, and it is the sum, the net sum, that dictates what happens to this one postsynaptic neuron. So let's say I have, in this case, what do I have? I have 5 presynaptic neurons. If they all fire together, and they're all of a similar magnitude, or they elicit the same change in graded potential, I have 2 that are inhibitory, 3 that are excitatory, so the net effect is excitatory. The net effect is excitatory. We don't know how excitatory. We know the net effect is excitatory. So that brings me to the 3 options, the 3 things that can happen. All of these incoming neurons converging on one postsynaptic neuron. Thousands, tens of thousands. 3 things can happen. 1, the net effect is excitatory, but it doesn't trigger an action potential. We don't reach threshold. 2, the net effect is excitatory, and we reach threshold. So we generate an action potential in the postsynaptic neuron, and that's sent down the axon. 3, the net effect is inhibitory. And there's no qualifier of whether we reach potential or not. If it's inhibitory, it brings it away from threshold. Membrane potential becomes more negative. So there's no qualifier there. Those are the 3 things that can happen. Now, I wanted to show you some of the complicated neural circuits, but we don't have time. So I'm going to stop it there. I know you're sad. You wanted to learn more. Let's leave it there. Have a wonderful Thanksgiving. See you in the morning, no, at noon on Tuesday. And good luck with the lab exam tonight.